Hi there. Welcome to today's PacBio webinar, Advancing Cardiology Research with Long-Read RNA Sequencing. Before we get started, I wanted to go over a few housekeeping items. First of all, the talks for today have been pre-recorded, and we will have live Q&A as noted below. So in this session, we will have Dr. Christoph Dietrich do the live Q&A. And then in the session later today, we will have Dr. Kimberly Aldinger do the Q&A. If you have any questions for specific speakers during the talk, go ahead and drop them in the questions module in your control panel, and questions will be addressed during the live Q&A. If you have a question for a speaker who is not present for the live Q&A, go ahead and send them and we'll forward it on to them. Please check out the handouts module for related PacBio literature, and then on-demand recordings for the session will be available at a later date. And then finally, we ask that you please fill out a really brief post-webinar survey. It helps us to generate content and find speakers that are relevant for you. Okay, so before we get to the speaker talks, I'd like to give a brief overview or introduction of the PacBio technology for full-length isoform sequencing, including the Iso-Seq method, which was leveraged by today's speakers, as well as our new Kinnex kits. And for this, I will be going offline. Okay. It's down here. All right. To start, let's talk about alternative splicing. Alternative splicing is a process where exons from the same gene combine in various ways, resulting in different but related transcript isoforms. These produce different proteins with different distinct structures and functions, and this diversity plays a key role in human biology and disease, with up to 95% of genes undergoing alternative splicing. So a classic example of where aberrant alternative splicing can lead to disease is in cancer with the BCL gene, where two different isoforms can have opposing effects. So one promotes apoptosis, while the other is anti-apoptotic and is overexpressed in cancer cells. So this is a really basic example showing that isoforms and not genes are the drivers of biology and disease. With short-read RNA sequencing, the cDNAs, which are shown in the gray bars at the bottom, are fragmented and then assembled computationally. And this is a problem since alternative splicing produces many isoforms that look similar, making it impossible to unambiguously infer the original set of transcript isoforms. In contrast, the PacBio Iso-Seq method sequences the full-length cDNA up to 10 kb with no assembly required, providing a complete view of the transcriptome at the isoform level. Recently, the Long-Read RNA-Seq Genome Annotation Assessment Project, or LR-GASP, consortium published a preprint on the comparison of different cDNA preparations and long-read sequencing platforms for RNA sequencing. They found that PacBio detected the greatest number of genes and had exclusively detected transcripts that were the longest and had significantly lower expression. Interestingly, they noted that Oxford Nanopore data, despite in this instance having comparatively more reads, did not consistently lead to more transcripts, indicating that the quality and length are important factors for transcript identification. They also found that while PacBio and ONT cDNA libraries both had good reproducibility and consistency across replicates, the consortium found that PacBio Iso-Seq method had twofold higher abundance resolution with the ability to quantify isoforms compared to the ONT cDNA data. And you can find the reference to the paper below. So as discussed in the LR-GASP paper, and which you'll see today actually in Christoph's talk, length and accuracy are both essential for single-cell RNA sequencing. On the left, the short reads, as I mentioned, have an incomplete view of isoforms, And other long-read technology has low accuracy, which may result in fewer usable reads. So only PacBio sequencing can provide full-length isoform and single-cell resolution with high accuracy. In previous years, single-cell Iso-Seq studies were limited in application because of throughput. And last year, we announced a method called MAS-Seq, which could be used to fully characterize a typical single-cell library with a single SMRT cell. MAS-Seq is a general amplicon concatenation method where short cDNA amplicons, like those produced by 10x Genomics Chromium single-cell platforms, can be concatenated to create a larger fragment to be sequenced full length with PacBio HiFi sequencing, thus greatly increasing the throughput. And then the reads are deconcatenated bioinformatically. So due to our accuracy, this concatenation increases the effective molecule yield without affecting the ability to call single-cell barcodes or UMIs. And in this publication from the Broad, not only was the long-read data able to recapitulate the short-read-based cell types, it also revealed isoform information with cell type specificity. So a few weeks ago at ASHG, we announced the launch of Kinnex kits for RNA sequencing. For single-cell transcriptomics, the Kinnex kit is an update to the existing MAS-Seq kit that I mentioned previously. This Kinnex kit is now compatible with both 10x Genomics Chromium single-cell 3' and 5' kits, and has up to 4-plex multiplexing at the library level now possible, which further increases the throughput. With the Kinnex single-cell RNA kit, you can now get around 6 million HiFi reads per Revio SMRT cell, which after deconcatenation results in a yield of roughly 8 million reads. For more information about this kit, I'm not going to go into the specs here. Please check out the handouts module, as I mentioned, and we have an app note with more information. So for bulk or full-length RNA sequencing, which Kim will discuss in her talk, we also leverage the same concatenation method that I described for the MAS-Seq kit to get higher throughput. So since the transcript lengths are typically shorter than HiFi long reads, concatenating cDNAs together means more transcripts per HiFi reads. And we'll see a preview of this Kinnex kit data in Kim's talk today. The workflow for this kit is similar to that of the single-cell workflow, but without the upfront 10x cDNA prep. With the Kinnex full-length RNA kit, you can now expect 15 million reads on a Sequel SMRT cell, or up to 40 million reads on the Revio SMRT cell. And multiplexing is also enabled here as well. And again, please check out the handouts module. We have an app note about this kit as well. I'll mention very briefly that for analysis, an end-to-end bioinformatics workflow is available on SmartLink. There's built-in support for human and mouse genomes with GenCode annotation available, or you can run the workflow without a reference genome for model organisms. At the end of this pipeline, gene and isoform count matrices are produced, which can be used with common third-party bioinformatics tools. And we have an app note, again, in the handouts describing such compatible tools. So also, if you'd like to take a look at the data yourself, please check out the datasets page of our website in the top right here, where you can find both full-length and single-cell Kinnex data. And very soon we will have the data that Kim is showing in today's talk actually also available. So in summary, the Kinnex kits provide full-length isoform resolution at an increase in throughput compared to previous PacBio Iso-Seq method. In the top figure, we're showing that Kinnex kits can capture long transcripts across a variety of species and tissues. In the bottom left, we show that you can get up to 16 times more data now compared to our previous Iso-Seq method on Sequel 2 when you switch over and use the Kinnex kits on Revio. Finally, with the increase in reads, we are starting to saturate known isoform detection So the other graph, we're showing here that roughly 80% of known isoforms detected with just 10 million reads across different samples can be done. And this is the expected throughput for if you are doing a multiplexing of 4 samples on a Revio SMRT Cell. So this increase in throughput allows you to capture isoform abundance, detect novel variants, and characterize differential transcript usage. So finally, in the next 2 talks, you're going to hear about how researchers are using the PacBio Iso-Seq method to better understand heart function, development, and disease. And with that, I'll jump back on during the live Q&A, or you can check out our website or reach out for more information.
Hello, everybody. Today I'm going to present our work on single cell and spatial transcriptomics using single molecule long reads with the multiplexed array sequencing approach on the PacBio HiFi platform. My name is Christoph Dieterich, and I'm presenting joint work that was performed together with Etienne Boileau at my institute here in Heidelberg, and also Claudia Quenau and Janine Altmüller at the Berlin Institute for Medical Systems Biology. So our background is, and where we come from actually, is from the Nanopore sequencing platform. So we were trying to basically bridge the gap that was existing for short read sequencing technology to the long read sequencing technology in single cell sequencing, for example, and spatial transcriptomics techniques with these 2 publications that you see here. So they are based on a hybrid sequencing approach using the Nanopore and Illumina short read sequencing to, on the one hand, study the entire transcriptome in single cells, and on the other hand, on the right-hand side here, to study the complexity of the transcriptome in slices of myocardial infarction from mouse models. So what you see here as a single cell sequencing application, and that's what we are mostly interested in, are cardiac myocytes that are derived from human-induced pluripotent stem cells. On the left-hand side, you see our differentiation protocol where we undergo a differentiation from day 0 to day 17 and check for cardiac markers. And all of that is described in a previous paper of ours in 2017. We applied a transgenic approach where we engineered a CRISPR-Cas protein, which is actually RNA targeting, so a Cas-Rx protein, into these stem cells, if you will. And this is a platform that can be used for many different purposes. So RNA splicing modulation, removing particular RNAs through cleavage, and so forth. So it's a whole plethora of opportunities that we have with the cell type. Of course, we wanted to know whether the cell type as such differs from the unedited background cell line, and if so, in what way. And that is why we set out to do single cell sequencing. So we did this initially, as I was telling you, on the Nanopore platform, combining in a hybrid approach single cell and single molecule full-length sequencing using for the long reads the Nanopore platform, but for the barcode identification, predominantly the Illumina sequencing platform. And the libraries were generated on the 10x genomic platform, then split into 2 arms, one for Illumina sequencing, one for Nanopore sequencing. And we wrote the software, and I don't have the time to explain to you all of the details that will bring these 2 parts together. And basically the Illumina sequencing serves to generate a whitelist of barcodes that were part of the library that was initially prepared. So the new MAS-Seq approach, which is actually available through Pacific Bioscience, caught our attention then because we wanted to think about ways to get rid of the hybrid sequencing and just use a single sequencing approach by itself. And the Pacific Bio platform enables us to do so because the high accuracy HiFi reads allow us to get rid of the Illumina sequencing part because the barcode identification is so much easier. So on the first part on the left-hand side, you see your standard protocol for generating a 10x library with a 3' end sequencing protocol. And then, of course, that is turned into a MAS-Seq library where you concatenate up to 16 folds cDNAs into a single hi-fi specific bioscience read. And then through various ways, then bioinformatically later, you regain essentially every individual cDNA, and that individual cDNA also carries a cellular barcode as well as a unique molecular identifier, all of which have been implemented through the sequencing protocol that you see here. So let me share a couple of results that we got from sequencing a mixture of the reference cell line together with the CRISPR engineered cell line, that one that would carry a Cas-RX protein. And we wanted to study how that would affect the transcriptome complexity and composition. So we started with a smart seq2 cell, essentially, and we obtained about 50 million reads. And then if you go through the bioinformatics pipeline, you get essentially individual full-length reads. You get the tagged reads that will tell you about the unique molecular barcode and the cell barcode. And then, of course, you refine further down your reads later, and that leads you to also some error corrections as being part of the bioinformatic pipeline. And then later, using the information from the individual text, you can deduplicate your reads. And you can see, I mean, following through this table here on the left-hand side, that we by and large regain as mapped reads after deduplication about 2/3 of the initial reads that we were generating. And about 500 cells are contained within this single cell experiment. So what does the data look like if we map it back of the reference genome? These are human cells, obviously, and here is the histogram of the mapped read lengths. The mean of the distribution is around 800 base pairs, and the median is a little bit below 718 base pairs. So if you wonder, what the transcript composition of individual cells are, we on average identify about 4,000 transcripts per cell in this data set. And with regards to the individual transcript counts per cell, we are ranging in the area of 14,000. The distributions are both shown here on the right-hand side. Now, how many different individual genes do we identify with that? So we are getting close to about 18,000. This would be the blue curve here at the bottom. And we get many of the isoforms, but they are typically also variations on individual gene loci. So the transcriptome assembly there is something that we were studying then in detail. And here's a kind of an overview of all the different structural categories that the individual transcripts that were reconstituted would fall into. So we have by and large 5' fragments. That is something that matches the reference sequence partially, but it gets truncated. And this is kind of the dominating number of transcripts that we see. We have alternative 3' fragments as well, where the splice sites are fully covered from the reference transcriptome, but there's an alternative 3' end. And then, I mean, you can just go through that and see what the different categories are. So if you wonder what the meta-transcriptome coverage would be, this is kind of the typical transcript body. Here printed out in percentiles from 5' to 3' end. And of course, since we are dealing with the 3' end library protocol, you see that the coverage is high on the 3' end and then slowly decays to the 5' end. But we still see a lot of coverage, so the bias is way less than you would get, of course, with a short read sequencing library on the 10x platform. So we hope to really recover the entire complexity of the transcriptome. So if you look at the individual transcripts, so this is not gene expression now, this is transcript expression, and you ask, I mean, what kind of cellular clusters you would observe. In a UMAP, for example, we ended up with recovering 3 individual clusters, shown here in colors, blue, orange, and green. And what is particularly interesting about these clusters is that they differ in the number of isoforms that get expressed per individual gene. The least number of isoforms, a little bit more than 1 per gene, gets expressed in this bluish cluster. Then we have a cluster where close to 2 isoforms get expressed. And then we have another cluster which is small in the number of cells, but also has a higher isoform per gene expression in comparison to the blue cluster. So after looking at additional cell markers and so forth, we think that the blue cluster are actually the ventricular cardiac myocytes, and the green cluster, for example, are more fibroblast-like cell fates. Now you, of course, are wondering how our Cas-expressing cells fall into this clustering, and that is what you see here on the left-hand side. So you essentially see that they are hardly distinguishable from the reference wild-type line. Cas-Rx expressing cells, they fall into basically all of these clusters, and it's really hard to discern differences. So by and large, we do think that both cell types are comparable, and the Cas expression doesn't change the transcriptome makeup considerably. So finishing this first part, we combine initially Oxford Nanopore sequencing in a hybrid approach with Illumina sequencing, developed a novel software tool to arrive at single-cell and full cDNA length resolution. However, we think that a better and non-hybrid approach is now to use the MAS-Seq protocol and the Pacific Bioscience platform, also with the latest invention like the Revio platform. And we do see that the performance on reference transcriptome looks fantastic, but many novel transcripts got discovered, and we are a little bit doubtful about the precision. So I think bioinformatics tool development is still necessary on that part. Okay, and finally, we could confirm that the wild type and Cas-Rx transgenes our cell lines are basically very similar in their transcriptome profiles. So let's move on to the next part, which will be spatial transcriptome sequencing. And here we focus on myocardial infarction. So you would think that single cell sequencing maybe answers a lot of these questions, but essentially, if you think about it, the original tissue context is lost if you destroy essentially the slices that you gain from heart samples, and if you put them into a single cell library. There have been other approaches like microdissection and so forth, but the same problems apply there too. So the characterization of cellular zones of injury and repair and remodeling is lacking a bit there. So our idea was to use the Visium platform, which is also a 10x genomics product, but that was relying mostly on short read so far, and I guess you can guess already what's coming next. So we essentially thought that it's very valuable to have long read sequencing because that would tell you something about the transcriptome compositions, and especially, for example, for model organisms that are not very well captured. It's very important if you want to identify novel protein coding genes and so forth, capture the entire transcriptome diversity to have long read sequencing. Okay, and that's what we did, again, with a hybrid approach initially. So we cut from myocardial infarcted hearts of mice. We cut tissue slices, put them on a 10x Visium, profiled them, generated libraries, also did in parallel the imaging and sequenced on the Illumina and the Nanopore. And that's what you get here on the right-hand side. You see here in the reddish part, the infarcted region, then you see in green, the border zone, and in blue, you see the remote area. So we profiled that in a lot of detail on the Nanopore platform. And then, of course, depending on the classifications, you could already see that for many genes, you identify many, many isoforms that can go up to 10 plus. And a similar picture, as I was discussing with you in the single cell sequencing, holds true there. So it seems that the remote zones, by and large, express more isoforms per gene in comparison to the border zone or the infarct zone. So our attention was caught by a particular class of transcripts that are associated with retained introns. Because we took notice that intron retention is a process that has been discussed previously in association with complex diseases. And there's very little known in the cardiovascular field about intron retention in that regard. And our statistical analysis identified that the odds were twice as great to find an intron retention event in an infarct region compared to remote regions with a significant p-value. Again, being intrigued by Pacific Bioscience technology, we thought that we can get rid of our hybrid approach and move on to the Pacific Bioscience platform. So our previous results on the nanopore with the gridiron were 6 million reads per slide, but essentially we lost half of them because of different various reasons, and the barcode assignment turned out to be difficult as well. So we try then the Pacific Bioscience MAS-Seq approach here on the existing Visium library, which we picked, which turned out to be best from our previous ones, and looked into it in more detail, sequenced it on the Sequel 2E, and basically that's what we got here. So we got 2.2 million hi-fi reads, and those were segmented into about 20 million full-length reads. And then, of course, the different quality criteria steps and processing steps were again carried out. And in the end, we end up with about 50% of the 20 million that could be mapped unambiguously to the genome reliably to reconstruct transcripts. And this is what it looks like. So on the left-hand side, you have, by and large, the entire counts. So the number of reads, if you will, per individual spot. This is only at near cell resolution because the Visium platform doesn't allow to resolve individual cells. But what you can nicely see is at the close to infarct regions, at the border zones, if you will, a lot more transcripts get expressed. This is here color-coded. The dark red one expressing spots here are essentially the ones with a lot of observations, i.e. read counts. And that is also then true if you just ask about transcripts. Of course, that translates directly into transcripts. That is also something that you can nicely see here on the right-hand side. Again, the transcriptome complexity is quite stunning. You see a number of recovered transcripts that agree with the reference annotation, about 14,500. But you also see a lot of novel splicing events in our data here, which show splice junctions which were not part of the reference annotation, for example. And just to orient you here, on the right-hand side, you have another figure legend that would explain what the different classes are. Again, I mean, doing the kind of statistics that we're interested in, so how many transcripts do we get per spot and how many observations do we have per spot, meaning like long reads. You see that the median is about 1,410 for the number of transcripts, and for the counts, we are close to 3,000. Again, I mean, mitochondrial read content is another interesting aspect of cardiovascular research because cardiomyocytes are the powerhouses of the heart, and they typically also express a great number of mitochondria, and that's also what you see that, you know, I mean, a lot of the reads also end up on the mitochondrial genome. On the right-hand side, again, you see a typical sketch saturation plots, how many genes were identified. It's close to 13,000, and then, of course, how they fall into unique isoforms. Now we were wondering whether we see characteristic isoform switching after myocardial infarction, and we could identify in total 117 of these events, and they fall into different classes. So if you, for example, compare the infarcted zone to the remote zone, you see that there's a different transcriptome usage using different isoforms. Here on the dot plot, you start to see this as well, depending on which region you're looking at. The isoform expression and abundance changes considerably for the individual genes. So a lot of cardiac genes are listed here. Markers for remodeling hypertrophy, like NPPA, for example. This can also be projected then on the respective images, such you would see how the expression pattern changes there. So very, very valuable information, a very rich information set. So then I would like to finish in summarizing the second part. So our software, SNAST, that was initially proposed for the hybrid approach, expands the spatial transcriptomic toolbox to use nanopore reads and short read sequencing. But with the Pacific Bio approach, we're gonna surpass that. And our example that I just gave you is essentially about the myocardial infarction model here in the mouse. Where we could explore the annotated and the de novo isoforms, which we were discovering here in much greater detail. Again, with PacBio, we were able to perform a non-hybrid approach, and we identified several isoform switches that are happening partially in clinical relevant loci, That is very interesting if you have a spatial heterogeneity in the heart and if you want to study that. So that is basically what we can enable and provide you with. So my last comment is essentially that the kit or the approach that I was presenting to you was all relying on MAS-Seq, and there's a new kit just recently coming out, which is called Kinnex, which of course provides you with even greater power. And so I can only encourage you to interrogate and ask your local sales representative about this one. With that, I would like to close, and I would like to acknowledge the people who did a lot of the experimental work. So that's Isabel Nama de Vries, doing a lot of the single cell sequencing, which I presented to you in the first part. Lucas generated the CASRX expressing iPS cell line, and she and Florian were providing samples for the myocardial function experiments. You'll find all of our software on our GitHub site, and if you're interested in joining our team, please feel free to do so. Thank you very much for your attention, and thank you for giving me the opportunity to present here.
Hello, my name is Kimberly Eldinger. I'm a principal investigator at the Center for Integrative Brain Research at Seattle Children's Research Institute and an assistant professor of pediatrics at the University of Washington. And today I'm going to talk about our work profiling dysregulation and cellular imbalances in Down syndrome. And specifically, I'm going to talk about the work that relates to the developing heart in this regard. So to introduce everybody to Down syndrome, in case you're not familiar, it is a systemic condition that results in many effects to many body systems. And so on the left-hand side is a schematic of all of the organs as well as the clinical manifestations that happen in individuals with Down syndrome. So Down syndrome as a clinical entity has been recognized for over 100, 200 years, but the appreciation of the molecular cause of Down syndrome has also been known for a long time, but not nearly to the same extent. So for about 50 years, we've recognized that an extra copy of chromosome 21 is what leads to all the clinical manifestations that occur in Down syndrome. And what we can see on the right-hand side is a karyotype indicating the extra copy of chromosome 21. So the clinical manifestations of Down syndrome have their origin within prenatal development, and there has been some research and investigation about the origin of many of these clinical symptoms relative to the early development of organs, but oftentimes these investigations are among very few individuals or are limited to a single organ system. And so what I've diagrammed on the right-hand side are just some of the organs that are relative to the clinical manifestations in postnatal individuals on the left with their clinical histopathology identifications within the prenatal period. And so we know that already during the prenatal period in individuals who have an extra copy of chromosome 21, that development of many organ systems are not occurring in the correct format. Our goal was to investigate the dysregulation and cell type representation among several different organs in prenatal development for trisomy 21. So we identified a series of specimens for cases that were verified to contain chromosome 21, as well as a set of specimens that were sex and age matched to the chromosome 21 cases. And we isolated nuclei and fixed those nuclei from 5 different organs, as well as skin fibroblasts, and we performed RNA-seq3. So this is a combinatorial indexing approach to investigating RNA expression in individual cell types. In total, we recovered 1.5 million single nucleus transcriptomes within our developmental atlas, which comprised 154 samples among the 6 tissues at the left-hand side, and within 76 specimens. We also performed a second set of experiments in order to corroborate some of the results that we found. And in this case, we identified additional specimens, fixed those tissues, the same that were represented in the single cell sequencing, and performed geomics in order to recover some spatially relevant RNA expression for a subset of the tissues and cases controls that we could evaluate. So congenital defects are very common in Down syndrome. In fact, up to 50% of individuals diagnosed with chromosome 21 often also have a congenital heart defect. And so what's schematized in the top panel are different presentations of heart defects, from an atrioseptal defect on the left-hand side all the way to hypoplastic left heart syndrome on the right-hand side, with increasing levels of severity. So ASDs are the most common congenital heart defects that are seen in Down syndrome, and improvements in surgical correction of the heart defects have led to an increase in survival and longevity for individuals with Down syndrome, but they still have ongoing health challenges related to these early developmental changes. And what's schematized underneath the pictures of the heart is a timeline for heart development in humans. And so what's important to appreciate is that the heart develops very early in the course of human development. And so that at the point in time where we have the opportunity to really investigate what is present or not present, what might be dysregulated in the hearts of individuals with Down syndrome, the heart is already completely fully formed. And so the specimens that we have access to allow us to look at mid-fetal gestation, which is between 10 and 20 weeks of development. And here by 9 weeks, the heart is completely formed. And so that gives us an opportunity to look at not the growth and development, but what is present in the physical heart at that stage. So for the purposes of this talk, I'm not going to talk about all of our results, but just about the results for the heart itself. And so we were able to include 24 samples, and we recovered almost 400,000 single nucleus transcriptomes among these samples to look at more depth and detail of the heart. And so the first thing that we did after filtering for mitochondrial percent and ambient RNA and doublets was to perform uniform map projections in order to identify the cells that are related to each other. And so we primarily used Seurat for our analysis and identified 26 cell types, where each of the dots in the UMAP on the left-hand side represents a cell's transcriptome and color-coded by the particular cell type, where there have been several recent reports of fetal heart development that facilitated our ability to identify what these cell types were. And so among the 26 cell types, ventricular cardiomyocytes were the most numerous that were recovered, while megakaryocytes were the smallest population that were retained here. We did look for differences in proportions of cell types between trisomy 21 and controls to determine whether or not there was an imbalance in the number of cells, and did not detect that. But when we instead look at gene expression, and in this case, we're using MAS-Seq and pseudo-bulking the individual cell types and then using DE-Seq in order to calculate gene expression. We can see in the heat map on the right-hand side where the gold bar, horizontal bar at the top is representing the trisomy 21 samples, and the bluish-greenish bar is representing the control samples, that there are clear distinctions in the RNA profiles among each of the cell types that are represented in trisomy 21. So even though there are not differences in the numbers of cells that are represented among the cell types, there are significant differences in the genes that are expressed within these cell types. And so vascular endothelial cells had the most number of genes that were differentially expressed, while the few neurons that are represented within a heart had the fewest number of genes that were differentially expressed. But what we don't appreciate from looking at the heat map, because there are so many genes, and so it wouldn't be helpful to put what the names of those genes were on the side of the heat map, we can instead look at a volcano plot and find some interesting results about what are those genes that are differentially expressed among each of the cell types that we've identified in the heart. And so what we find is that there is this sort of suspicious player, SUN, which is a gene located on chromosome 21. And so it might be expected to play a role within Down syndrome, but that among the 26 cell types, SUN is the gene that is differentially expressed most frequently within the cell types. And this actually extends to every tissue. And so I'm not showing those results, but if we were to take our gene expression results for heart or for lung, we similarly find that SUN is the gene that is differentially expressed in the majority of cell types throughout the body. And this really caught our attention and we thought was quite interesting. And one of the reasons why we thought it was quite interesting is that we could identify it also within tissue sections. And so I'm not showing expression of the tissue sections, but this was our validation using nanostrings geomics in order to look at histological RNA representation within sections. And so shown in the second panels are sections of the heart, one trisomy 21 and one control, and then the zoomed in regions of interest that we were able to capture. So what we can see in the volcano plot on the right-hand side, that again, among the genes that show increased expression, SUN is among those genes. So even in more of a bulk format, we're able to capture expression of this particular gene. It's not just limited to the individual cell types. But SUN itself is a very interesting gene, and it's a DNA and RNA binding protein. As I mentioned, it is on chromosome 21, which is relevant for trisomy 21 or Down syndrome in particular. It's a ubiquitously expressed nuclear protein and splicing cofactor, and it promotes splicing of cell cycle and DNA repair transcripts. It also binds to DNA sequences, and more recently, heterozygous de novo variants cause a severe neurodevelopmental disorder when they occur within this gene. But other than that, there is not a lot of information about the connection between this particular gene and Down syndrome or within heart development itself. But what it led us to ask the question is whether or not there are transcript isoform and usage differences in the trisomy 21 hearts. And to be able to address this question, we were excited to have the opportunity to use the new Kinnex library prep protocol in order to obtain isoform and transcript level information that is really not able to capture in short read RNA sequencing, especially at the single cell level in its own assay. And so we selected additional cases, isolated total RNA, and then we're able to access the Kinnex library prep in sequence across PacBio. So instead of predicting any differences in transcript and isoforms, we could directly investigate what might be happening in this regard. And as you can see in the table below the workflow, that we were able to obtain more than 6 million reads for each of the 4 samples that were sequenced, that the majority of these reads aligned, and that 31 to almost 40 million transcripts were obtained among these 4 samples. And so these are really exciting data that we're looking at in a very preliminary way now, and are very excited to look at in more depth and detail, as this is our first experience analyzing any long-read sequencing data. So the first thing that we did after being able to use FLAIR to generate a count matrix for both the genes as well as the isoform transcripts that are present within the long-read data, we again use DE-seq like we did for our single-cell RNA sequencing analysis to identify what the genes are that are differentially expressed. And what we found was that the genes that we identified were in fact distinct from what we had appreciated within our single-cell analysis. And we're really excited about the possibility of repeating some of our single-cell analysis and then layering on the long-read sequencing in future experiments. And so because we only consider 2 cases and 2 controls, we are not well powered for a lot of investigation, but we have some really exciting preliminary data. And so because there were so many different genes, 440 in total, that we identified as dysregulated within our single-cell RNA, we looked a little bit less conservatively with the long-read expression data, and were able to identify 12 genes that were dysregulated in both cases. But we're also interested in the 64 genes that showed differential expression in the long-read format that were not present among our single-cell data set. And so also interestingly, the majority of the genes that we identified, in fact, were not present on chromosome 21, and we're really interested to look at that further. But what's even more exciting than identifying different genes, that some similarities and some differences, is that there were more transcripts relative to genes that were differentially expressed. And so we identified 141 differentially expressed transcripts, and these were among 33 different genes, again, with fewer of those genes being represented on chromosome 21. And then just looking at the combination of genes that resulted from either the long-read differential gene expression, the transcript expression, or our single-cell analysis, there are certainly some overlaps as well as some distinctions. And so these allow us to look at more depth and detail into the biology, both with future and more thorough analysis in characterizing these genes, as well as pursuing additional assays. So overall, of what I've described is that we've generated a multi-tissue cell atlas of Down syndrome in early development, and that in the trisomy 21 heart, we do not detect proportional differences in cell types, but we do detect significant gene dysregulation that impacts both chromosome 21 genes as well as genes that are in other chromosomes of the genome. We're really excited to pursue SUN further and think that that might be a key regulator in Down syndrome development, not only within the context of the developing heart, but within many of the organ systems that are affected in this condition. And we're very excited to delve further into the Kinnex results in order to be able to link the differences we see in SUN to the isoform differences that we are potentially identifying in our preliminary data. And with that, I would like to thank in particular Ian Phelps, whose picture is listed on this screen, who has done really the lion's share of work for this project, and to Lindsay Seidnor, who has been jumping into the deep end with learning the analysis for the long read sequencing. And with that, I'll take any questions.
All right. Thanks again. Very exciting talk. So as a reminder, Christoph will be doing the live Q&A for today's session, for this particular session. Thanks again for joining, Christoph. This is really exciting work. We've got a few questions in the chat. So I guess I'll start with, we had a few questions around your use of Visium Spatial transcriptomics. Can you talk a bit more about how that was done? Was it done directly by applying the MAS-Seq protocol to the cDNA library post Visium? Any tips and tricks for how you did this?
Yeah, sure. Thank you very much for putting this question forward. So basically, indeed, so it's the same kind of streamlined workflow. So you just apply the 10x protocol, once for single cell, once for Visium, and then turn this into a MAS-Seq library by using the respective kit. So there's no magic involved. Yeah.
Great to hear. Anyone can do it. Another question from the audience. Is it expected to get that high of a ratio of novel, not in catalog isoforms with spatial long read sequencing?
Mm-hmm. I mean, it was a little bit difficult to tell about expectations because, I mean, the only comparison that we do have is essentially either using short read technology, which have certain caveats, as you all know, or comparing it to our Nanopore project. And in the Nanopore project, we also see a lot of novel splicing sites emerging there as well. So, I mean, I cannot give you a precise percentage, but the overall profiles look similar there too. So I would suggest that this is something that can be independently corroborated. Yeah.
Okay. Let's see. I have a question while we wait if there's any more. So in regards to that Nanopore Illumina hybrid approach, did you do a comparison with PacBio in terms of the usable mapped reads as well as the results you get in terms of like number of genes or isoforms identified?
Yes, we did indeed. So in terms of the usable mapped reads with a Nanopore approach, we are very much limited because we have to identify unambiguously the barcodes and the UMIs, and that was really difficult. So we put a lot of efforts in the software, but in the end, I mean, in comparison, if you just talk about the single cell data, so 2/3 of the cDNA reads could be used for PacBio and only 1/3 for the Nanopore.
Great.
Yeah.
Okay. Let's see. Sorry, some are coming in. It's a good general question. Were you able to identify any of the novel isoforms? And I guess I had the same questions. How do you validate novel isoforms, isoform switches, and just what does that mean biologically in this case?
Yeah, so it's a great question. Actually, we are more like a computational lab, but indeed we took courage and identified, validated a couple of these isoforms also by PCR. For example, the PDLM5 gene was validated. There's an isoform switch in the myocardial infarcted heart, and that is what we could validate there. But I mean, we didn't go into high numbers.
Let's see. I had a question actually about the intron retention. Did you observe intron retention to the same extent using your hybrid approach? And then, you know, in terms of validation, do you think that these retained introns are functional or do they undergo nonsense-mediated decay?
Yeah, so in terms of the intron retentions, I mean, we see basically the same thing. So the Nanopore also supports this. And in terms of the functional consequences, for most of the intron containing or retaining transcripts, you would expect a nonsense-mediated RNA decay event to happen. But that is something that we didn't study in detail. Yeah.
Okay.
Yeah.
All right. There were a few questions about the Kinnex kit. Maybe I can answer. One was, do we have an idea of whether the Kinnex kit affects the median or mean length of transcripts recovered? They're concerned about a bias towards shorter transcripts being retained through the concatenation. I will say it does not affect the mean and median length of transcripts recovered. If you take a look in the handouts at the app note for full-length Kinnex kit, we actually have some data showing this. It's the same as well for single cell, but we don't have the data in the app note. So please take a look at the app note. And then somebody asked, let's see. someone was asking about analysis. Christoph mentioned walking through the Iso-Seq pipeline. Was that pretty standard for you, and did that work well with other tools that you had previously used?
Yeah, so I mean, so we tried, of course, previous versions also of open source packages to deal with MAS-Seq data, and I mean the really big breakthrough was to use the PacBio software itself, because I mean the previous versions are of course less well maintained, I would say, and more difficult to handle. So the PacBio software is really convenient, and it's also super fast. Just in comparison, if some of you know, for example, the Longbow pipeline from MIT, that gave us not the same quality of results, let's say, and a lot more waiting. Yeah.
Great. Yeah, and if you have more, we have put a bioinformatics app note. You can also check isoseq.how, which has some documentation. It's also available in our SmartLink pipeline. We're glad to work here. It was pretty easy to use for you. So now we have a few minutes for Q&A, and I will field some for Kim. So I know you just got your data back, but just wondering, have you been able to do any follow-up work since you got the Kinnex results, particularly in the Sun gene that you could share?
Yeah, thank you, Nina, for that question. We're really excited to look at Sun even further. Some of what we've done so far with these data beyond what was in the presentation is to look more at Sun in detail, because Sun is known to have significant splicing differences in and of itself. And so we're still investigating that. We're also looking into the potential targets of Sun and whether or not there's any enrichment and differences in the isoform expression of the targets that Sun is known to interact with too.
Cool, great. A question I had, do you see overexpressed isoforms or even genes in other chromosomes when it comes to trisomy 21, and just what is the hypothesis behind that, and how does that affect your phenotype?
Yeah, we do. So first and foremost, we see just in any RNA sequencing data set, we see a 1.5-fold increased expression in chromosome 21 genes. And we use that as a QC metric for any of our RNA-seq analyses and make sure that that's present before actually delving into further analyses. But you know, having an extra copy of a chromosome is really a genomic disorder. And so we're really interested to look at what the more global changes are that are happening throughout the genome. There are some exciting data sets that are with Hi-C and for other genomic methods too, that I think intersecting some of those results with what we have here will help us unravel some of those non-chromosome 21 genomic details as well.
Great. Looking forward to seeing that work. I think we have a question I can try to answer. Could PacBio generate total RNA sequencing library or only poly-A selection? We do total RNA, so our input is 300 nanograms. RIN score greater than 7 is ideal. You could optionally do poly-A selection. You can also do other types of depletion if you want as optional steps, but we do generate total RNA. And then maybe just a final question for you, Kim. Since you mentioned you're pretty new to long read, how are you thinking about, gene level centric analysis to isoform level analysis? Are there any unexpected findings or changes in your analysis plan that you need to think about?
Yeah, so I think it's really exciting to think about the potential of the long reads, and I'm really glad that there are products that are available to us to do this now. We've spent a lot of time in the short read and poly-A space, and we know that we're even missing some gene expression, let alone isoforms in general. And even as you've mentioned with your example of BRCA in the introduction to this webinar, that there are significant differences in isoforms, and in trisomy 21 in particular, when we have some genes that we know regulate isoform splicing, we're really excited about
investigating those possibilities and what their potential implications are for disease mechanisms.
Great. So unfortunately, we are at time, so I'm going to wrap up. Thanks again for Kim for joining us. Thanks to all for attending. Just want to close up with a few things. So in the next week or so, you're going to get an email with a link to this recording. If you want to go back and listen to any part of it, we'll also have Christoph's Q&A on demand as well. Second, please keep up to date on any webinars by visiting our homepage and checking out pacb.com/events. And then finally, again, we're going to have a short questionnaire afterwards. It takes about 2 minutes and it really helps us understand your needs and plan for future webinars. So thanks again for joining. Hope to see you again in a future webinar. Have a great day.